cell plants that are being used extensively. There's a need to conserve crop genetic diversity. You can see here the seeds of the same plant, the different kinds of seeds here. Now, we need to preserve them because narrow genetic base can be disastrous. There have been instances like potato famine and the maize problem in the US where the entire germplasm was lost. So we need to conserve them for our genetic improvement programs as well as for posterity and use them for the benefit of mankind. Biodiversity generates economic value, extractable plant products, compounds, genes and species to meet the industry needs. 40% of the global economy is based on biological products and processes. So you can see how important these resources are in terms of economy across the world. Biological resources are going to be important for future welfare and security of nations. Now coming to a problem that we see very often these days. Young boys and girls who obtain their degrees look for jobs. Now this is one picture from Allahabad Employment Exchange which I could find. Now they have standing in queues to register themselves to seek a job. Now India's job growth, the jobless growth needs to be stopped. We need to create 150 million jobs this decade. There are reforms to labor laws and policies to develop the skills and infrastructure. And one would see that the present government has taken very strong steps, serious steps to create more jobs so that our youth can be employed gainfully. What one would observe that large number of students go for interviews and only one would be picked up, or sometimes even that does not happen. So what we need to do, and I'm now bringing to you a different thing with regard to bioeconomy. See, natural economy was there in early part of 1900 or before that, where people were producing things in a narrow village or a community, exchanging it with others. There was no concept of selling them. There were kind of exchanges. Then gradually we moved towards better economies. Fossil economy, as you know, has been there for such a long time. Now, fossil fuels are diminishing. And what we need to do, we have to improve our economic development, use the bioresources. And as I said, bioeconomy is the answer for the future generations. What is this bioeconomy? Refers to the set of economic activities relating to the invention, development, production, and use of biological products and processes. Bioeconomy has been there because of the necessity that people felt. Due to population growth that is taking place so rampant across the world, by 2030 there will be need for 50% more food, 30% more water, and 45% more energy. People are forced to find alternatives to non-renewable raw material. Climate change is an imminent reality. The scarcity of non-renewable raw material, etc. These are the things which are leading us to utilize these bioresources for the economic benefits of our people. Now, bioeconomy, from it we can get most of the biomass. From the biomass, we can get fine chemicals, medicines, food, chemicals, bioplastics, transport fuels, electricity, and heat. Now, if you look at the bioeconomy of our country, you will find it is more than where we aim to achieve 100 billion bioeconomy. How that can be done, what we have to utilize towards this meeting, towards this end. Flower power. The international floriculture trade stands at 45 billion at this time. Indian floriculture industry heralds a new beginning. We have given a big slogan, million jobs with $1 billion. Now, if you look at the floriculture export in 2017 and 18, it was 78 million US dollars. So there is a lot of potential here. I bring to you some beautiful orchids from Northeast. I'm not going to tell you the details of these. Just have a look at them. So beautiful, highly floriferous. They have long shelf life. These are lady slippers, as they are commonly called. Again, some rare orchids from the region Northeast. Now, we have tried to multiply them in large numbers and provided them the saplings, the in vitro raised plants, to the women folk in Meghalaya who have been able to grow them in their backyard, and they are generating revenues. You can see these orchids growing in their fields. The other aspect, which I can tell you from my experience from Biotech Park Lucknow, they are producing very high quality banana plants, and these have been provided to farmers of Uttar Pradesh. And the genetic engineering and other related techniques can help produce different kinds of plants. As you can see, this is such a big banana that has been produced in Taiwan, and there are 3,600 bananas in one bunch. I don't know whether this can be done on a large scale or not, but then the possibilities do exist. Then we talk of herbal products. Global market stands at 75 billion, showing a 7% annual growth rate. India's share is just 2.5%, which is one-fourth that of China. Now, if you look in the American markets, on the shelves you'll find most of these things coming from China. I think there is an opportunity for us to bring in, to export more of such things globally. The global nutraceutical industry stands at 385 billion US dollars, which is again a very important area which we can explore. International herbal medicine market to reach 5 trillion by 2050, showing annual growth rate of 10 to 20% as per World Bank report. A strong Indian pharmaceutical industry would provide affordable medicine at competitive price to a large number of people. And as you can see, there are a number of products that are produced in our country. Now, just to give you a glimpse of medicinal and aromatic plants which are grown in backyards of villagers in Northeast or even in forests, there is very rich diversity. As I show you now, this is the fruit morphology and different types of chilies that you find in Northeast. And this is one such chili, which is called Gujalakia, has very high pungency, more than million Scoville unit. And this is found in Babylon and other parts of Northeast. And we have tried to multiply this in large numbers, at the same time tried to culture them and enhance the production of capsicum, the active principle. Now you can see that even pepper guns are produced. Women folk use these small little lipsticks which contain this compound, which can deter somebody who is coming to rob. Then talking about bamboo mission. World market for bamboo stands at 29 billion, and our bamboo mission is expected to provide 8.6 million jobs. There is market worth rupees 6,500 crore with just an investment of rupees 2,600 crore. And in this area, I'm very happy to say we have been able to make big leaps. India contributes. On the other hand, the thing that we need to consider is that India contributes only 4% share of global market. The largest area, which is 11.4 million hectares, is under bamboo cultivation. Now, more than 100 species of bamboo are found. Uh, the third area which I chose to talk to you is about Startup India, Stand Up India. Learn more and be partners. Why is it important? We have the bioresources, then how we should use it. Now, there has to be a passion in our youth to take this challenge. Here, I show you the Pad Man. Now, it is Lakshmikant Chauhan. This was a movie which was made, and this gentleman, Arnachalam Muruganathan, received a Padma Shri for the work that he did. He has produced sanitary pads for women folk at 1/3 of the cost of commercial pads. He has established machines in 23 states of our country and planning to expand it to different nations. He, looking at his family members and friends, he chose that he should take up this task, and hats off to such people who have made immense contribution towards the problems that we face. Now, even an Oscar award was given in 2019 on a small, short film period. Now, here also, the women of Indian villages who band together to make menstrual pads. Now, we have to think in a different way. Not that everyone is looking at one direction. You have to see from all sides what is possible and look for the best. There has to be thinking out of the box. You can see egg cartons have been made of molten pulp for decades. But then there are lots of concepts out there that one must try. Now, wheels were known to people for thousands of years, and only in 1970-72 they were put to suitcases by the Americans. Now, earlier it was like this, and one of the pilots found that carrying suitcase, wheeling this way was difficult, so he invented 360-degree suitcase with 4 wheels and a handle here. Now, these are the things that we need to see. Our ancestral wealth shows that there is so much that we can pick up from there and use it for the benefit of mankind. Now, a very simple concept, as I can show you here, same tea sold differently. You have different ingredients added to them: tulsi green tea, tulsi honey chamomile, pomegranate, and so on. So, by adding adjuvants to the product, you can make a difference. Even the same juice packed differently. Now, as you can see, that a team of youngsters who are working as software engineers, one of them used to bring panna from his home, and one day while sitting at lunch table, they decided, why not bring it to the people? And they went to Mr. Narayan Murthy and said that, can you fund us for this program? And it was funded, and now you have paper boat. The food processing, lot of jackfruit in Northeast getting wasted, bamboo shoots, etc. So there is a need to process and this food not only for people in our country as well as for export purposes. Freeze-dried fruits. The fruit production in our country has increased now. Therefore, there is a need to preserve it and use it. What we need to do now, boost bioeconomy knowledge in schools, colleges, and universities. Agriculture and the agri-food industry to fisheries, forestry, biorefineries, chemistry, and bioenergy has yet to enter the public consciousness. People must know that we have very rich bioresources in our country. I've been talking only about plant bioresources because I'm a student of botany, but there are animal bioresources also which can be used by people across the country. Boosting bioeconomy knowledge in schools. In the schools, we must tell our students, the youngsters, that how rich is our biodiversity, what properties these plants possess, and integrate the subject of translational research into academic courses. I can tell you there are courses in many countries, MSc in bioeconomy, biotechnology and law, interdisciplinary programs where biology, economy, law, and marketing is taught. So we need to develop new capabilities and skills. We must organize seminars and lectures for our students. And I think that this forum is providing the right kind of environment to reach out to the youngsters of our country. Now let us see the objectives of skill development. We need to support startups and help in entrepreneurship development. Educational programs to create awareness about entrepreneurial and job opportunities. At the same time, try to help enable our farmers to increase their productivity through biotechnological interventions. As I showed you earlier, tissue culture raised orchids, banana, and large number of such things can be done to help people. Now, what India has done in this regard, India's efforts at promoting entrepreneurship and innovation. We have a Startup India program that promotes entrepreneurship by mentoring, nurturing, and facilitating startups throughout their life cycle. Make in India, designed to transform India into a global design and manufacturing hub. Atal Innovation Mission to promote a culture of innovation and entrepreneurship. Then support to training and employment programs for women. And there are many other programs like Biotechnology Industry Research Assistance Council funded programs, which is a not-for-profit public sector enterprise set up by DBT to strengthen and empower emerging biotechnology enterprises. Stand Up India, which is for underprivileged, mainly women entrepreneurs. Trade-related entrepreneurship assistance and development programs. Pradhan Mantri Kaushal Vikas Yojana, which is doing marvelously well for creating livelihood opportunities for people. National Skill Development Mission, Science for Equity, Empowerment and Development, web, social media platforms for entrepreneurs. And then let us talk about women as biotech entrepreneurs. India is third largest startup hub in the world. But in 2016, only 9% of the startup founders were women. Now, this is something which we need to change. Involve more women in entrepreneurship. Out of 1,022 biotech companies in the last 5 years, 33% were only owned by women entrepreneurs. Women entrepreneurs are really very successful because they have life sciences appeal and they have innate connection to life science, to nature. Now, there are 300 to 500 new startups coming up each year, and we expect that 2,000 biotech startups would be there soon. There is a need to have gender gap to close. Closure of gender gap could lead to 6.8% gain in GDP. Now we know that 50% of our population is women, close to 50%. So we should not leave them behind because they can be very important contributors in national growth. Women's equality in India could boost its GDP by 0.7 trillion by 2025 or 16% as compared to the business as usual scenario. Index of women entrepreneurs ranked 52 out of 57 surveyed countries. So there is a need to change this. Women participation in the workforce has unfortunately decreased from 35% to 26% in 2018. I'm talking about this as a general, not in the area of biotechnology. Harnessing the full potential of women entrepreneurs can promote innovation, economic growth, and job creation. Now, here I present to you some successful stories where women have contributed immensely. Ekia Biochemicals, Arda Chandra Mauli, and Gayatri Thankachi, manufacturing of biotechnological, biochemical, and enzymatic products and microbial plant growth promoters. Here you have Sarah D'Souza, who has a company called BioCells Technologies to produce quality analysis, microbial testing, biochemical analysis, and stability testing. Then Anuradha Acharya, founder and CEO of Osmum Biosolutions. Uh, she has been awarded the Young Global Leader by the World Economic Forum in 2011. And then the most famous women entrepreneur of our country, Kiran Majumdar Shaw, Indian billionaire entrepreneur, chairperson and managing director of Biocon Limited. They are involved in industrial enzymes, biopharmaceuticals, deal in number of drugs including diabetes, oncology, autoimmune diseases, and so on. Awarded the Otker Gold Medal in 2014, Financial Times Top 50 Women in Business, most important women entrepreneur from our country. Now, how the students can benefit by the support that is being provided by different agencies. Department of Biotechnology provides seed funding through Biotechnology Ignition Grant, BIG through BIRAC, then support for entrepreneurial and managerial development of SMEs through incubators, public-private partnership, seed funding, equity-based with co-investment from private investors, seed funding, and so on. Support from DBT in the form of through BIRAC, they are planning to devote a corpus more than 100 to 200 crores for its bioequity funding. So funding is available, bioresources are available, youth is there, and it is that they are requested to use them for the benefit of our country. And I would here bring at the end Prime Minister's appeal, skill development of the new generation is the foundation for Atmanirbhar Bharat. So transforming the dreams into reality of our youth, we need to do all this, and I hope that I have been able to convey to you some of my thoughts that I thought are important for our young boys and girls across the country. Thank you very much.
Thank you very much.
Is some organizer there?
Am I audible, sir? I'm here. This is Dr. Aditi from NBRI. Thank you very much, Professor Tandon.
Do we have some organizers? Yeah.
Hello. Yes, sir. Am I audible? Thank you very much, Professor Tandon, for your highly intriguing and very, very informative talk. I'm sure it has...
I can't hear you. Maybe you can turn off your picture.
Yes.
And then...
Hello. Sir, can you hear me now? No, I am unmuted. Hello? Hello? उसमें से कर लीजिए. Hello?
Hello? Yes, I have concluded my lecture. Yes.
Hello?
Hello?
Can you hear me now?
Yes, please. I have concluded my lecture.
Hello. Sorry, sir, for the... future scientists and current scientists and entrepreneurs. So I thank you from the bottom of our hearts for your talk, and we hope to hear you more in the future. Thank you so much, sir.
Thank you.
So now I request Dr. Manish Bhoyar to please introduce and welcome the next speaker of our today's event. Over to you, Dr. Bhoyar.
Hello. Good morning to all. Thank you, Dr. Aditi. Lichenology Society at CSR National Botanical Research Institute, Lucknow. Dr. Upreti joined as an acting director, CSR National Botanical Research Institute, Lucknow, on 1st June 2016. Dr. Upreti, after obtaining master's degree in botany from Kumaon University, Nainital, has pursued PhD from the Lucknow University under the guidance of Dr. D.D. Awasthi, and joined CSR India as a senior research fellow in the year 1982 and scientist in the year 1988. Dr. Upreti served as a chief scientist in a senior national CSR National Botanical Research Institute, Lucknow, prior to his joining as an acting director. Dr. Upreti is well known for his research work in the lichen taxonomy, biomonitoring, climate change, and bioprospections. In his career, he received several awards such as JBS Halden Memorial Scroll, Professor B.A. Razi Medal Award, Professor K.S. Thind Award, Environmental Conservation Award, Award of Excellence, and currently he is a Fellow of National Academy of Science, India, and Fellow of Society of Ethnobotanists. For his outstanding contribution in the area of lichen taxonomy, he was awarded the prestigious E.K. Janki Ammal National Award for Plant Taxonomy by the Ministry of Environment, Forest and Climate Change, Government of India. He was the member of 11th Indian Expedition of Antarctica in the year 1999 and 92. Dr. Upreti has discovered more than 100 lichen species new to science, together with addition of more than 200 lichen species in environmental lichen species. Then in environmental pollution monitoring, climate change, bioterrorization, bioprospection of lichen for development of antimicrobial nanomaterial. He has published more than 350 research papers in reputed national and international journals, and authored and co-authored 13 books, 42 book chapters, and 20 research articles. Dr. Upreti is a nodal scientist for CSR for 5-year supra-institutional network project and coordinated 11 biological science laboratories of CSR. He is principal investigator of more than 35 projects funded by GST, DBT, ISRO, CSR, and guided 35 PhD and 43 MSc MPhil students. Dr. Upreti is a chairman and member of several committees at national and international level and bilateral research projects with Germany, Chicago, South Korea, Ukraine, and Russia. He is the president of Indian Lichenology Society and honorary member of International Association of Lichenology. With this introduction, I will just welcome to Dr. Upreti for his talk. Now it's over to you, sir.
Namaskar, Dr. Manish, thanks a lot for good words you have used for me. It's really an honor for me to deliver this lecture, and I thank all the organizers of Vigyan Sarvatra Pujyate-Festival. I have worked on lichens for the last 45 years. There is some technical problem. I think I will share my slides and I will start it. Just wait. Is it visible?
It is visible now, sir, but not in the presentation mode.
Is it visible?
Yes, sir, it is visible.
Okay, so I was talking about that. These lichens, they have an ability that they can grow on any substrate which is perennial, whether it is man-made or it is natural. So they can grow on trees, they can grow on twigs, they can grow on barren rocks, they can grow on even leaves or on the mosses. So depending on their type of substratum, we can classify them into different groups. Again, some growth forms are there, means on the basis of their growth forms, particularly sometimes they form a crust, sometimes they appear leafy, and sometimes they are thread-like. So depending on their growth forms, we can divide them into different groups too. If you see, as I told you about that, it is the lichenological researches in India. They were initiated long back. The Britishers or natural lovers in British Army, they collected lichens from different regions of our country, particularly the Himalayas and South Indian regions and Eastern Himalayan regions, and they collected those samples and they sent it to the European countries. So most of these material which was earlier collected by the Britishers, they are not available in India. And it was my honorable teacher, Dr. D.D. Awasthi, who established a school of lichenology at Lucknow University Botany Department in the 60s of the last century. He started working systematically on lichens, and he started revising different groups or compiling different informations which were carried out or published by the Britishers, and he published a lot of research work on lichens. Recently, it is CSR NDRI who collected represented different localities of our country, whether they are protected areas, whether they are state-wise, and we are in position that in the last about 50 years, we are in position to tell something about the diversity of lichenology in India. Dr. D.D. Awasthi, my honorable teacher, is here in the picture. You can see he established a school of lichenology in Lucknow University, Lucknow. But unfortunately, after his superannuation, all his specimens, all the lichen specimens which he has collected, his personal herbarium, and even his personal literature, it is all transfers to NDRI. And NDRI is taking care of his literature, taking care of his specimens on a permanent loan basis. If you can see the fast pace of lichenical researches took place starting from 1960 to this up to 2020. We have more than 100 areas. Dr. Awasthi. Excuse me, slides are visible? Can somebody
Yes,
tell
sir,
me?
they are.
Okay. This is about the lichenology.
Well, they are visible, but they are not in presentation mode, so it will not move. Yeah, it has to be in the presentation mode to move.
Okay.
So now it is in presentation mode, sir.
Okay, there is some technical problem here. Anyway, it's okay. How the light-critical researchers to the fast pace of research is to establish research centers in different places with our help, whether it is universities or organizations, and we more than 100 places are there where these light-critical researches are going on, and the students, they are working on different aspects of lichens in our country. So I very quickly tell how we identified lichens. When we go to the field, it is their reproductive organs. When we see, or on the basis of their reproductive organs, we can segregate them, and I mean some reproductive organs, they are button-like. Sometimes they are linear. Sometimes they are raised, pedicellate. So depending on their reproductive organs, we easily identify or segregate them into different groups. Even we can go for the morphology, what happens in case of lower group of plants. If you compare the taxonomic characters, you have few taxonomic characters as compared to the higher plants. So the taxonomy of lower group of plants is starting from biophytes or to lichens or algae. We have few morphological characters or anatomical characters. So we largely depend on chemistry or other aspects of these plants. So as earlier I told you that it is not a single plant, lichens are composite plants or symbiotic association of algae and fungi. So lichens, they have mostly green alga within them, or the lichenization or lichen formation involves mostly the green algae, and within this green algae, it is the Triboxia type of alga which are most frequently involved in lichenization. So mostly in 80% of lichens, it is green alga, and only 10 or 20% lichens, it is the blue-green alga or the cyanobacteria involved in lichen formation. As I told you, if this is about the anatomical structures of reproductive structures, again they play important role in taxonomical studies or in segregation of different taxa. Even the ascus tip, sometimes this ascus, which is only of a size of about 10 to 15 mm, it has some starch, and when we put iodine on the sections, these particular structures, they form different shapes of this color, blue color, and on the basis of this structure, some genera are differentiated. So thus, the taxonomy of this group is very complicated, or it is very difficult, because it is based on some micromorphological structures or anatomical structures. You can see the tips of this ascus and the color of how the iodine reacts with it, and we segregate them into different groups. Lichen, symbiotic association between algae and fungi. The vital photosynthetic reaction is going there in alga. And depending on the type of alga, whether it is green or blue-green, they produce glucose, retrol, levital, sorbitol. These are the carbohydrates, and they passes it to the fungi. Now, throughout the world, 20,000 species of lichens are known. Out of these 20,000 species of lichens, more than 150 species of alga and three major fungi, that is Ascomycetes, Basidiomycetes, and sometimes the Deuteromycetes type of fungi, they are involved. So depending on the type of fungi and the carbohydrate it receives from algae, there are three biosynthetic pathways by which more than 1,500 secondary metabolites of lichens are known from all the 20,000 species of lichens so far known from the world. Out of these 1,500 secondary metabolites, 150 only are common with other type of or other chemicals which are known from other group of plants. 1,350 are unique. They are not produced by any other group of plants except they are produced only by lichens. And they belong to these dipsides, dipsons, dipsides. So these three pathways, it is started with acetate polyvinylate pathway, malabonic acid pathway, and cyclamic acid pathway. There are three these biosynthetic pathways which are responsible for production of lichen secondary metabolites, which are not known in any other group of organisms. So how we identify them? We identify them with some color tests. We can even identify with this myelinating crystals, or sometimes we use this TLC, and few means if we have to go for identification of a few chemicals, we use SPTLC or SPLC, even GCA and GCMS, or the most sophisticated techniques for analytical analysis of these chemicals. So spot test, even if you remove the upper part of the lichen, you put some chemical, that is particularly we use K, potassium hydroxide 10%, then it changes. If certain chemicals are present in that lichen, the color will change. It will change like yellow or red. So we can suppose that if it is color, is it changing K plus red? Yes, we can tell yes if this lichen contains this type of chemicals or dibenzofuranons or dipsidones. So depending on the color which it produces on its surface, we with certain chemicals, whether it is PD, K is potassium hydroxide 5%, 10%, PD is paraphenylene diamine, and it is again a organic compound, and it also gives different stains with lichen thalli when we put it on it. So microcrystals, we all know that certain chemicals they produce crystals, and with the shape and the structure of these crystals we can identify the chemicals. After the advent of these molecular tools and techniques within lichens, there is a sea change in lichen taxonomy nowadays. I will tell you some example of one Parmelia, which is most frequently known lichen in India, and earlier it was known with Parmelia species. After the advent and application of these anatomical some structures and some micromorphological structures and supplementation with molecular tools, one that Parmelia genus is segregated into 30 different genera now. So there is a lot of role played by these molecular techniques, and there is a lot of scope still to work on Indian lichens, applying these molecular tools and techniques. We can easily divide our country, India, into three parts: tropical, temperate, and alpine region. We all know that the altitude from sea level to 1,500 meters is tropical. We have a special type of plants growing in this area, and you can see the most common one is Soria robusta. So Soria robusta harbors more than 70 species of lichens. It is most suitable plant which can provide a suitable substratum to lichens to colonize on it, because it provides different niches starting from the base. The base, well, sometimes the roots they are buttress, they are coming out at the on the ground, and sometimes the lower trunk is laden with soil. So it gives different niches, if you move upwards, the upper trunk is more less smooth. It gives substratum for different type of lichens to colonize. Even the twigs, they have different type of lichens. So a single Sodia robusta tree can harbor more than 70 species of lichens to grow on. These are particularly tropical lichens. Mostly it is dominated by parmelioid lichen taxa. Temperate region, we all know that this is between the altitude from 1,500 meters to 3,000 meters. In India, this particular altitude is more diverse and has a rich diversity of trees, and when different variety of trees are available, so lichens of different groups, they colonize on different type of trees. So we all know that it is quercus. In Western Himalayas, quercus trees or oak trees are very common, and even there are 4 or 5 species of oak are there in Western Himalaya, and they are colonized by lichens. At lower altitudes, quercus leucotrichophora is very common, and it has luxuriant growth of lichens. In the middle and higher altitudes, it is Quercus semicarpifolia, or we can call it black oak. It is an excellent habitat for lichens to colonize. We have mentioned about 90 species of lichens. They are growing on Quercus semicarpifolia. Next comes there are certain regions, or alpine regions are there, or in India we have some special areas, we call them cool deserts. They occur at an altitude around 4,000, 3,000 above or 4,000 meters. So alpine region comes from 3,000 or above. Here you will not find big trees or some trees which are planted, whether it is salix or poplars. And in lower altitudes, sometimes mostly the coniferous trees are there, and trees of this Bitula, Bhojpatra, they are there. And on higher altitudes, particularly if you go to Ladakh area, if you go to Gangotri, more higher places, you have plenty of these rock felts over there along the rivers. You have boulders, so you will not find trees there, but you will find a special type of lichens growing on rocks. You can see that these lichens, they are very dark in color. Why they are dark in color? Because they have to develop certain pigments. These pigments act as a screen to protect the plant from high UV. Because lichens, they are growing in high altitudes on exposed rocks, so they have to develop certain chemicals or pigments to protect their alga from high UV. As I told you, if you see the statistics of lichen diversity throughout the world, yes, more than 20,000 species of lichens are known from the whole world. If you see the history of lichenology in India, in the year 1965, we have only mention of 900 species from our country. But you can see the fast pace of lichenological researches. In 2020, we have 3-4 growth of lichen species. Means so much work has been carried out in different centers of our country, whether it is NBRI, whether it is ARI, or different organizations and different universities throughout India, and we have more than 3,000 species of lichens are known from our country. Again, it is very interesting that we have a large number of endemic species, these such species, they are only known from our country. They are about 17% from this area. And we all talk about megadiversity countries. Megadiversity countries are 17 throughout the world, and India is one of them. So we have more than 10% of lichen, they are occurring in our country. So lichens are very, if you see, our country has rich diversity. No country has such diversity. If you go to the Himalayas, you have lefty Himalayan peaks are there. But on the other side, you have sea. On east, if you go, you have world's highest rainfed area. But on the west, you have deserts. So you have a variety of climatic conditions and topography. So you have a different type of plants, including these lichens. We worked on some aspects of conservation of lichens in India. We mentioned that there are certain protected areas. Protected areas, we know that we have some biosphere reserves. More than 19 biosphere reserves are there in our country. But then we have plenty of these national parks. Then we have wildlife sanctuaries. So we emphasize to work on biosphere reserves. So we have accounts of lichens, whether it is Amarkantak Achanak Mar Biosphere Reserve in Madhya Pradesh and Chhattisgarh area. Then we have Nanda Devi Biosphere Reserve. Then we worked on lichens which are growing in Jim Corbett Research Area, where this tiger project is going on. So we declared certain places or certain areas as lichen sanctuaries. Even we are successful in creation of one lichen garden, which is an initiative which we have, with the help of Forest Department of Uttarakhand, we established a lichen garden in Munshari area. So once we have this idea of lichen taxonomy, idea of word diversity, then we explored this plant in different aspects, or its different aspects, multi-dimensional approaches of lichenological researches. So you can see that even we can predict the health of forest, even we can tell the age of forest with help of lichens which are growing in and around that forest. Then we can carry out a large number of environmental pollution monitoring studies with lichens, which is well known. Then even we worked on some climate change and to predict the glacier retreat in some areas of our country. As I told you, lichens, they have unique chemicals, so there is a lot of scopes to work or to exploit or to explore their pharmaceutical potential, whether it is applying, utilizing this nanotechnological techniques. So I will tell something about those aspects which we have worked, and our institute has worked a lot on this area. Biodeterioration. Biodeterioration means how, when in city areas or in man-made artifacts, when the lichens, they do not find a place to grow on trees, they have an ability that they can grow on man-made substrates. And this particular type of study is called biodeterioration, means how lichens, they're growing on monuments or historical buildings, and they cause damage to their upper surface. If you see within Lucknow, you can see that there is a large number of monuments. India is again culturally rich. In North, we have different type of monuments. In South, we have different type of monuments. Mostly the dome-shaped monuments are here in Northern India. But if you go to the South India, we have flat type of these monuments. So depending on the architectural pattern, depending on the construction material, or what type of rocks are there, whether it is a sandstone, whether it is a marble, so lichens, they grow on them. Now there is a debate whether they damage the substrate or whether they protect it. There is a great debate in European countries, but they say no. These lichens, when they are growing on a monument, they give an aesthetic sense. But in our country, being a tropical country, sometimes they form black patches and it gives a shabby look. And there is a need that there are some agencies, government agencies are there, which are responsible for conservation of these monuments in our country. We all know that it is Archaeological Survey of India. So when they face a problem, they approach and they tell us whether these lichens, they are damaging or what is the extent of damage. Then we tell something about the extent of damage scientifically, systematically, because it depends on the type of lichen which is growing on it, whether it is a lichen which is producing certain secondary metabolites and tightly attached to the substratum. So the nature of lichens and the nature of chemical it is producing, depending on that, it needs conservation or it needs removal or its conservation. We all know about succession, plant succession, and it is the Custos lichen which is pioneer in succession, because it has an ability that it can grow on a barren rock and certain chemicals are present inside it. It secretes those chemicals. Those chemicals are biodegrading, or the chemicals they produce, they break the calcium or they break the magnesium inorganic salt present in the substratum and make a loose crust of soil. So lichens, they have this phytogenetic significance, and in plant succession, whether it is primary succession or secondary succession, lichens, they play a vital role. How this activity is going on? I was talking about biogeochemical activity. Lichens, they have a number of chemicals. Lichen chemicals are unique because they have too many functional sites. They have polar groups and they are easily dissolved in water. These polar groups are nothing, CHO, COOH, OH groups. Once they are dissolved in water, what they do, you can see that this calcium is present in the rocks or in the substratum. And this is one ectramorine, a lichen substance. This is very common occurring in lichens. You can see that OH, COH, they are the functional sites. What they do, one pair of the electrons seems to be provided by oxygen or carboxyl of the aldehyde group CHO. It and OH, what they do, the calcium within its vicinity, it attaches that calcium and the metal and form a metal complex. Now this ectramorine is with calcium, and what it does, after detaching calcium from the substratum, it liberates it into free state, and it again assumes its normal form. So this is we call chelation, where one organic substance react with one inorganic salt or form a metal complex and liberate it into free state. So sometimes you can see that lichens, where lichens are growing, there is high accumulation of calcium in and around of those lichen patches in nature. There is biophysiological activity also. Some lichens means depending on the type of environmental condition. If there is more moisture, the lichen thallus, which is tightly attached to the substratum, expands. When there is dry season, what happens? This lichen thalli, it curls up and it breaks the rock particles which are attached with it. And this is very slow process, and indefinitely it is going on from years. Thus, how these lichens, they are involved in succession, in phytogenetic succession, phytogenetic significance they have, means they are involved in breaking the rocks and making soil or making suitable substratum for other group of lichens. Then it is colonized by bryophytes, colonized by pteridophytes to come and colonize. As I was talking about that, how this Archaeological Survey of India, what type of information they need, we provide them that what type of lichens are growing on this monument. Sometimes these lichens, they act as a cloth. They can protect and they can reduce the temperature to the building or to the substratum. But if the lichens, they are tightly attached and they are producing chemicals, secondary metabolites, they are more effective deterrent, and they have to, means they can be removed. There are different techniques by which we can remove lichens from their monuments, or from these monuments, and the chemicals particularly, they belongs to this ammonia solution, and it should not have that much of concentration that it can damage the monuments. So particularly the fine sculptures, they are removed from lichens through this ammonia solution. Now, bioprospection, how we can use lichens, and first comes the medicinally important lichens. The leaves which are available on, whether it is from ethnic groups, whether it is from folklore, whether it is pharmacopoeias, throughout the world only information on 500 species of lichens are known, which are medicinally important. Out of these 20,000, so there is again a lot of scope to work on these aspects or the medicinal potential of lichens. And even if we see in India, we have only 160 species of lichens on which we have worked for their medicinal potential. The leads which so far available, as I was talking earlier about the shall push, which is mentioned in Rig Veda, it is the first mention of Ausadi in that literature, and it was lichens. So whether it is old literature, whether it is pharmacopoeias, or whether ethnic usage of lichens, we all consolidated on this information, and we have worked on these aspects too. We have listed about 60 species of lichens from India which are used or which have potential as medicinal in medicinal in curing different diseases, whether it is fungal or it has. You can see that again various diseases which can be cured by lichens. Lichens can be used as antiseptic, antifungal. So you can see that recent years we have anti-inflammatory, anti-cancer activities. They are associated with lichens because lichens, they produce a large number of these secondary metabolites. But there is a drawback. Why these plants, they are not exploited or explored so far in greater extent for their medicinal potential, because if you go to the nature, they are their biomass is very low and they are very slow growing. Sometimes the lichens, they assume a diameter of I'm audible. Hello.
Yes, sir. Yes, sir.
You are audible.
Okay. And this slides, they are moving.
Yes, sir.
Okay.
Yes, sir.
Okay. So if you see that a large number of diseases, about 40, or they can be there where we can be used these lichen compounds for their treatment. So based on those leads, within our group, along with Dr. Vigyan Singh and these chemistry persons, we worked on development of nanomaterials, antimicrobials from lichens. We have leads about this Asnia longissima that it was used in various skin diseases earlier. So there we treated it against Candida albicans. We all know that Candida albicans is treated with, or the ointment which is available in market is fluconazole. It's a broad spectrum antifungal. It is used. But Candida albicans is in three different growth forms. It may be viral, it may be virulent, it may be sapromycial, or it may be mycelial one. So if it is in mycelial form, it can easily be controlled with this ointment which is available in market. However, if it is virulent or sapromycial, it cannot be controlled with this ointment which is available in market. As I told you, lichens, they have very functional sites, and they are best material or best chemicals to develop nanomaterials. So what we have done, we have isolated one quercetin, it's a flavonoid from Asnia longissima from nature. We collected it, then we taken out this, or we isolated this quercetin, we characterized it, sterilized it, and we developed the nanomaterials from this quercetin. And the nanomaterials from fluconazole, which is available in market. What happens when we synergize these two nanomaterials and we put, or we treated against this Candida albicans, it controlled all the three growth forms, and the efficacy is very high, is more than 200 times better than the fluconazole which is available in market. What happens there? The quercetin, it enhances the production of furcinol, which is the compound responsible for inhibiting the growth of Candida albicans, which is present in this ointment which is available in market. So this is how the synergy between two nanomaterials, which is available in market and which is available in nature, can control or can be used as developing such type of pharmaceuticals. Nowadays, earlier it was thought that lichens, they are only symbiotic association between algae and fungi. No, there are a lot of bacteria, there are a lot of of saccharomyces fungi, they are also involved in lichenization. Apart from them, there are again some fungi which are living inside the lichen thalli. We call them endolichenic fungi. And they are excellent source of a number of alkaloids, polyketoids, steroids, even xanthones. So here there is a need of one chemist. There is a need of networking between a biotechnologist. Well, only after identifying a lichen, identifying its endolichenic fungi, the culture of endolichenic fungi is very fast, or it is quick, or even there are techniques that we can manipulate the growth of this endolichenic fungi in cultured conditions. And one chemist can tell about the metabolics, or about the interaction, or about the chemoinformatics of these chemicals, and then a biotechnologist can tell something about the pathways, or how we can transform the gene of this particular chemical producing endolichenic fungi into a organism to produce good biomass of, or huge biomass of these chemicals. So this is an area which needs networking, and is a future scope for lichenologists and chemists, even biotechnologists to come together and in a networking and explore lichens, because India is very rich in such type of lichens which can be used for their pharmaceutical potential. You can see the structure of some lichens that, again, I have told you that they have certain polar groups, they have uniqueness in their compounds, they have too many functional groups, easy to develop nanomaterials. If you come to see in India, every year 1,000 tons of lichens they have collected either from Himalayas or from South India. As I told you that the Hyderabadi biryani, which is famous for its taste, the fragrance in that Hyderabadi biryani comes from one Parmelia, Parmotima reticulatum, which is luxuriantly growing in South India. You can see huge bags of this lichen, full bags of lichens in around the forest, sometimes which can be commercially exploited for this Hyderabadi biryani. You can see this is the view. There is segregation of lichens is going on. Grading we can show. We can tell you the first grade, second grade, third grade, and fourth grade. First grade, only 2 species of lichens are there. They are Evernia stem species, Nepalensis, then Parmotrema reticulatum. Then a huge waste of these lichens, it goes to Kannauj. Kannauj, you know, this is a place in Uttar Pradesh famous for its perfumes, and one perfume Hina Atar is prepared from lichens there. So for conservational point of view, can we culture lichens in laboratory? So we started that particular culturing lichens in laboratory conditions. We know that in lichens, it is the fungus, or it is the fungal reproductive organs, they only involved in sexual reproduction. There is no involvement of this alga. So the fungal sexual reproductive organs, apothecia or perithecia, they bear fungal spores. And these fungal spores can be taken out in a petri dish, because due to this moisture, what we do, we take a petri dish with some culture on it, and we put one fruiting body at the top. And due to this moisture, what happens, this fruiting body bursts and it liberates the spores on the petri dish, and we can see that the growth of this mycelium or microbiome is very fast. The microbiome produces sometimes the same secondary metabolite which is present in nature, but most of the time what happens, sometimes it doesn't produce the same secondary metabolite which is present in nature. Even we can grow them, or we can get the bulk big biomass of this lichen microbiome, which has a secondary metabolite similar to the nature in rock culture also. We were talking about this lichen in perfumery. As I told you, there is a huge amount of lichens. It is commercially exploited for making this hina atar, And we have worked on this aspect that what are the different species which are exploited, and we have mentioned that it is about 40 species which are knowingly, unknowingly exploited for making perfumery in our country. So Kannauj, what they do, mostly the parabolic lichens, or which are bigger in shape and size, they just boil it and they pass that steam to the sandal oil. And the lichens, they have certain resinoids, and the perfume is fixed on that resinoid. So we called it Hina, and it is very costly, and it is sold at cost of about 5 lakh liters. So very costly perfume. Lichen compounds, they have unique characteristics too. They have an ability that they can color a protein. So mostly the wool and silk, it is colored with, it is not colored with synthetic dyes, but it is the lichen dye which easily, or it is the lichen compounds which easily changes the color of silk, or particularly the animal originated fibers, silk, wool, or even from the fiber which we get from rabbits. So what we do, the ammonia-rich aqueous solution of lichens means we dip the lichens in ammonia-rich solution. Due to this fermentation, lichen substances, I told you, dipside or dipside dizones, they hydrolyze and they change into oxalic acid. Then we covered it with the lid for a few days. So due to this carbon decarbonization, this oxalic acid converts into orcinol, and this orcinol is when reacts with nitrogen from ammonia in the solution, it changes into orcine. This orcine is a derivative, and depending on the type of pH or moderant which we put, whether we put some oxalic acid or some citric acid or some salt, there are changes in their color. So different lichens are responsible for producing different colors, and since we have received a DST project to work on in these aspects, so for conservation point of view, we started culturing lichens, or only the lichens which are producing some dyes, and can we go for culturing such lichens in laboratory conditions and yield the color? Yeah, we were successful, and we have used these lichens, we cultured them, and we used the culture for conservation point of view, so that in nature the lichens, they should be not exploited for this particular technique, or for this technique or particular aspect. So we used this fiber. You can see F1, it is the controlled one with the fiber which we obtained from Banaras silk, and C, D, E are different natural one, the dyes which we extracted from nature one, but even you see on the label G1, this is all the dyes which we got from cultured mycelium. Lichens, they are very unique plant, and they have unique morphology, and they are best known for their pollution or environmental pollution monitoring studies. What are their characteristics? They do not have the root system as in hydro plants. Whole of the lower surface is attached to the substratum, and there is no cuticle on the upper surface. So what happens? Whatever atmospheric fallout is present in atmosphere, or whatever metals are present in the substratum, they are easily absorbed by lichen thalli. Then they are very slow growing. Means some lichens which are growing on rocks near glaciers, they have a growth rate of 1 millimeter in 100 years. So such type of their nature or their characteristics can be used in climate change or glacier retreat studies. Then even the lichens after orchids are very sensitive to microclimatic changes. Means we know about this. Orchids are very sensitive to microclimatic changes or atmospheric moisture in a particular place. Similarly, the lichens are thereafter. Orchids, they are considered as most sensitive to microclimatic changes. Lichens, we know that if a forest is intact, virgin, undisturbed, it will definitely have more moisture, more shade. So different type of lichens will grow within it. If it is disturbed one, more light, more wind will pass through this forest, different type of lichens will grow over there. So depending on the type of lichens which are growing in a particular forest, we can predict the condition or the health of that forest. If we have certain lichens which loves to grow only in moist condition are plenty in that particular area, we can say that this forest is intact, is a virgin one or not disturbed one. Even we know that it is the tufstose lichens, they come and they first colonize on trees or on twigs. Then, because they have an ability that they can grow on a high pH, means around 6 or 5 or 6 pH, if that twig has, that particular twig has. But what happens? It is replaced by foliose one when the pH changes. pH goes toward alkaline one, 7 or 8, 6 or 7. So, depending on or what is the type of lichens, whether that forest is dominated with climax stage of lichens, we can say that how much time it will take. Means a forest is whether 200 years old, 100 years old, it can be predicted based on the type of lichens growing on that particular forest. So depending on communities, lichen communities, which are growing on a particular forest, we have mentioned about 16 such lichen binder communities in our country, which are associated with the condition of the forest. For example, this calcicoid lichens, if it are growing there, and then we can say that this forest is an old growth forest. It has an ecological continuity. If some ethnoid type of lichens are growing over there, yes, we can say that this forest or this particular condition forest is devoid of acidic gases or it is healthy. So we have published on those communities, bioindicator communities, which India has. There are about 22. Lichens, they are known as best indicators because they are very sensitive to the acidic gases. Again, they are true indicators because they very quickly tell you the changes, whether it is morphological, anatomical, or physiological, when there is a smallest aspect of pollution in that particular area. They are excellent accumulators. Certain metals, particularly chromium, cadmium, nickel, which is lethal for a group of organisms or plants, they are beyond their physiological need. They are accumulated within lichens, and lichens, they do not die. Sometimes 50,000 ppm of copper, 50,000 ppm of other metals, they are estimated from lichens which are growing on some such type of rocks which are rich in calcium or such metals. We all know that within our cities there is no lichen growth. One study which is published in Nature in 60s, which what they have done, they have studied that in London, in UK, the areas which has rich growth of lichens, they do not have lung cancer patients. But the areas which has less number of lichen communities or lichen diversity, they have more number of lung cancer patients. So directly, if you have plenty of lichens in your area, yes, it is healthier region. If less, it is polluted area. So we all know that within city areas it is difficult to get the lichens. So this is the method we call it transplant technique. And in this method, what we do, we go to the nearby healthy area. For example, in tropical areas, we have plenty of these mango orchards where you can find lichens growing, and you can take out lichens along with their substratum, paste them on boards, hang those boards in different chaurahas at different height, on 4 feet and 10 feet, because certain metals which are coming out from vascular activity, some are lighter, they liberate or they disperse higher, some goes to the ground. So you can have measurement of different, or they accumulate within lichen thalli. As I told you that lichens, again, one unique nature of lichen is they fulfill their water need from atmospheric moisture vapors. See, they do not suck water from the substratum. They fulfill their need from the atmospheric water. So in atmospheric moisture is, if some metals or some pollutants are there, they easily absorbed or adsorbed in the lichen thalli because there is no cuticle. So they directly goes to the intercellular spaces between the mycelium, or sometimes if they enter, they go to the cell inside. Then lichen zone mapping, yes, we can map a bigger area. I will tell you something about this. Next slide, I think, is related with this one. Then sampling individual lichen species, measurement of contaminants accumulated within a thallus. So this is particularly picture the studies which we have carried out in Lucknow. We divided Lucknow whole district into 1 kilometer grid, and we take Hazratganj in the center of this one. So from Hazratganj towards 30 kilometers, we move north, then south, and east and west. And within that 1 kilometer grid, we just saw whether lichens are present or not present, or if present, what type of lichens are there. And then we have a zone. In the center 9 kilometers, we do not have any lichen growth. Means we consider is lichen desert. This is critically a highly polluted one. So lichen desert, critically air pollution is there. Out-struggle zone, yes, some lichens are there, but they are toxic tolerant lichens, so medium air pollution is there. Then comes the transitional zone where we can see some growth of foliose lichens, it is low air pollution. Then comes the outskirts of the city, where we do not, we find normal growth of lichens, we find all type of lichens, so very low air pollution we can correlate with it. Such type of studies are carried out or can easily carried out by students or the beginners who wants to pursue researches on this plant. Detailed mapping and listing. If we have some old records, if somebody has mentioned that this area or the flora of lichens of that particular area, for example, we have got a published account of about 53 species of lichens from Kolkata city, as known as was in the year 1860. So we again revisited those locations. Even Botanical Garden of Calcutta Botanical Garden, it was possible that we again revisited the same tree, tried to locate that lichen, whether that is growing over there. Then we saw that there is complete change in lichen community within that, and it is the urbanization which is responsible for decline of lichen diversity. Lichen has an ability that they can accumulate metal, metalloids, organic, inorganic, even radionuclides. And we have studied their potential to biomarker potential too. Means they, as I told you, they very quickly show changes within their this separate mechanism, within their physiology. Even we have studied that they can very quickly show damage, DNA damage within that plant, which we expose for different concentration of chromium and cadmium, which we transplanted in our garden. We all know about pH, polycyclic aromatic hydrocarbons. These are particularly the carcinogenic compounds, pollutants, which are liberated when there is very high vascular activity. So, in our country, in Himalayas particularly, during this pilgrimage through Badrinath, you can see a large number of vehicles they fly on that area. So we studied what is the level of pH in different places. Certain pH, polycyclic aromatic hydrocarbons, they may be 2-ring, 3-ring, 4-ring, and 6-ring. 6 rings, they are heavier molecular weight, and they are accumulated at the valleys. And the lighter one, 2 and 3 pH polycyclic aromatic hydrocarbons, they are dispersed higher, and sometimes they are found at the top of the ridges. So we depending on the type of pH present, we can say that which area is more polluted, whether it is valley or it is the tops. And even we have proposed a proxy regarding this polycyclic aromatic hydrocarbons, which we produced at place. They moved from this place to Himalayas, and we have given the proxy that in 2013 the landslide in Kedarnath area, it may be due to the pH which were accumulated there in that area, or pH which has lighter molecular weight, and they move from these plains to Himalayas, responsible for that cloud burst in Kedarnath area. Lichens, they can accumulate a large number of inorganic metals, but if a smallest speck of acidic gases, whether it is SO2 or NOx, NOx or SOx gases are present in the atmosphere, you will not find lichens growing over there. Depending on the type of pH, if it is towards lower pH, means around 5-6, then you can see the symptoms. We all know that the sulfur SO2 is very much soluble in water when there is moisture condition. It forms an acid H2SO3, which further breaks H plus HSO3 ions. We all know this. H is reducing. It reduces the magnesium present in the center of the chlorophyll and convert this chlorophyll A into pheophytin, a brown pigment. So what happens? And liberated the magnesium. So what happens? The process of photosynthesis, it eases and then this lichen, they're showing symptoms, morphological symptoms, as well as anatomical systems means due to this pollution. If we see that depending on the air pollution sensitivity of lichens, what we consider crustose lichens are the least sensitive, or we can consider them as tolerant. And foliar, they are moderate, and fruticose are very sensitive. So if there is a pollution in a particular area, first we will see that we will not find any of these fruticose and polycose lichens. But crustose lichens, which are growing on rock, they are more tolerant. We consider them as more tolerant. I was talking about this lichen variety, that lichens, they are very slowing, and if they are growing near an glacier on rocks, they assume a diameter of 1 millimeter in 1,000 years. So you can see that these orbicular structures, if its diameter is 3 millimeter, means yes, it was that rock on which it was growing, maybe exposed long back. So such type of this data we have used, and we studied the climate, this glacier retreat in Himalayas, in certain glaciers, eastern and western one. This is an example of how we studied this glacier retreat in Pindari glacier area with the help of lichens. We divided a distance of 1 kilometer starting from the 0 point, from the glacier snout, where today is the glacier snout. Then away from this 1 kilometer, we took diameter of this rhizocarbon geographicum, the lichen which form the circular structure, after an interval of 50 meters up to the glacier snout. So we have a correlation starting from 1 kilometer to 0 kilometer. So what we predicted, or what we calculated, that since this lichen is growing 1 millimeter in 1,000 years, so this glacier, if the diameter at 1 kilometer is bigger, so we calculated that this glacier retreated 1 kilometer in 675 years. So such type of studies, when we published, it was Indian Science Research, Indian Space Research Organization, who were responsible for studying this climate change studies in Himalaya. They approached us, and we have worked in collaboration with Space Research, Indian Space Research Organization of Ahmedabad. Even we predicted that the glacier retreat is more faster in eastern Himalayan region than the western one. Climate change, we all know that it is the temperature, it is the precipitation which is responsible for that climate change or shift of the species. And even the gases or the green gases which we produce. So what happens when there is shift, or when there is change in a particular species from one place to other space, due to climate, due to change in precipitation, due to change in temperature, or due to these gases? it starts climbing or it's moving towards upwards. When it shifts or it climbs, it needs acclimatization. For acclimatization, it has to change its morphological or phenotypical changes. And we all know that it is the mRNAs which are involved in this particular activity when a virus gets its acclimatization. So lichens which are growing or which are moving to the upper part, when their rocks are exposed, they are facing a lot of sun rays, so they have to develop certain chemicals, particularly the anthraquinones, which protect its algae from high UV. So this migration and this acclimatization long last means when this to acclimatize in a particular place, it influences its cellular activities too, means particular of that plant. We all know that if it influences the cellular activities, there is a change of mode of reproduction. And if there is a change of mode of reproduction, entire biology of the species change and change in the compounds. So it leads to the genetic changes. It took thousands of years, but you can easily see the changes or acclimatization of lichens or adaptation of lichens in harsh conditions. The symbiotic association enable lichens to grow on any difficult situation or in environment or fragile environment. So we can even find lichens growing in Antarctica. I will tell something about the Antarctic lichens too. We worked on Mars. This is microspring lichen acids, and particularly the cyanolichens or lichens which contains this blue-green algae. They produce this type of amino acids, and they have an ability that they can quench the UV effect in those plants. And these plants or these lichens are best suitable for development of these anti-UV creams in future. There is a lot of scope in this particular type of lichens to work on that. Not only these compounds, but the secondary metabolites, when there is an increase in altitude, an increase of UV, there is increase in quantity of certain secondary metabolites too. So along with this Indian Space Research Organization, we worked on this global warming using lichens. Means we all know that after 70 years, definitely the average temperature will rise. So if it rises 2 degrees centigrade, can we create that temperature which will come 70 years in our glacier or in our alpine regions? So we use this open top chamber. These open top chambers are simple. They are 6 hexagonal sheets and covered with one transparent sheet. And there is a sensor in the center which tells you about the temperature, increased temperature, or which is recorded throughout the year. So we have compared, one is the control one. We have covered this land with stones, and other is covered with this open top chamber. You can see the changes, and even you can see the changes within the physiology or biochemistry of the plants or lichens which are growing inside that open top chamber. This is how we work on lichens. With help of lichens, we are studying this change in climate change studies in alpine regions of India, in all the 5 states of our country, and we have this multi-summit approach. What we will do there at the altitude of 3,000 meters, 3,500 meters, and 4,000 meters, we put these sensors which will tell you about the temperature and precipitation at 4 places at an altitude of 3,000 meters, 4,000 meters. And they all are simulated, or you can see that connected with satellite. And a person who is sitting at Ahmedabad can see at 56 places how the temperature and precipitation is changing within an interval of 1 hour, and they have already developed the spectra of flora which is growing on that area within 5 meters, within 10 meters, and they will continuously watch it for 20 years, the coming 20 years, that how it is changing with climate change, when there is change in precipitation, climate change. Earlier, they have established a satellite, such type of studies, to see uh, the climate change, or we know that the tree line is only restricted at 3,000 meters. After 3,000 meters, you will not find any trees. So it is mostly the grasslands, and then comes the firma snow, and then glaciers. So we studied that, or India studied that in the last even 40 years, the tree line it moves forward 40 meters, means 10 meters per year. That is how the glaciers they are melting, how the permanent snow is melting, and the grass cover is increasing, and tree line is moving towards the glaciers. So it is the, but at that time they do not put these sensors regarding temperature and precipitation. Now such studies are going on, and they will last long last for next 20 years. Sometimes when we collect specimens from field, we keep them in herbarium, and if we put correctly record the altitude, latitude, even the GPS data on it, such type of information are useful carrying out a long-term monitoring program in future. For example, we have collection of lichens from Darjeeling area of Eastern Himalaya, which was collected in 1970 and mentioned about the localities. We again revisited those areas, and we again taken out the climate change studies, and we have calculated that there is a great change in communities because there is a great change in precipitation and temperature of those areas. So we have published these accounts too in good publication here. Now we will move for longitudinal studies which we have carried out in Antarctica. It was NPRI started participating in Antarctic research in the year 1991-92 when I had been there as a summer participants, because there are two type of participants are there. I will tell to the students that there is a lot of scopes to work in Antarctica, to visit Antarctica, because every year they have they ask for different proposals to work in Antarctica, and there is one institute which is located at Goa, NACOR, which comes under the Ministry of Earth. They are responsible for conducting this Antarctic expedition together with the Arctic expedition. So I will tell you about how you can apply for that. But we started in the year 91-92. After me, Dr. Vigyan Pandey has visited after 5 years. Then Dr. Sanjeeva Naika visited there for twice after an interval of 5 years. Then we have one student participation also from our institutes to Antarctica. So what we have carried out there, we have worked on the lichen diversity of that region. We have worked out the metal or pollutants accumulated on lichens of that region. We have worked on how these lichens they go for their adaptation strategy in Antarctica. So these aspects we have worked over there. If we tell something about Antarctica, yes, we sometimes called it South Pole of Earth, and it is below 66-degree south latitude, Antarctic Circle we called it. It is 14 million square kilometer, means larger than India and China if we put together, and 97% is covered with ice. Whenever we talk about Antarctica, we think that, oh, there is no land mass or there is no exposed rock over there, but it is there. It is not totally covered with snow, 97% is only, but there are certain rocks or these nunataks, we call them, exposed rocks are there which are not covered by snow. And lichens, braphites, they grow on those ones. Then 2.5% ice-free areas are near coast and in Antarctic Peninsula. 90% of world's ice is there in Antarctica, 75% world supply of fresh water from there. So we started Indian Antarctic expeditions. They were started by it's a vision of our late Prime Minister Indira Gandhi, and the first expedition, it was organized by Department of Ocean Development, and its name was Operation Gangotri because Gangotri is a holy place in our India, from where this our holy river Ganga is originated. Similarly, just named that place Dakshin Gangotri, they have given to this one, and they have put this Operation Gangotri to visit Antarctica, and it was Professor Dr. S.J. Kasim was the member of 21 participants who visited Antarctica in 81-82. So it was a journey of 33 days covering 11,000 kilometers, and so they have established a base camp. It was on the coastal regions of that area. If we talk about who owns Antarctica, there is a large number of history that it started in the 70s when James Cook and Scott they visited that site. So whoever visited in Antarctica, they have their territorial claims over there. So mostly where we stand, it is a Norwegian claim, and you know that it's the year 1959. It is what's called an International Geophysical Year. After that, anybody can visit Antarctica for peaceful researches. We cannot establish a defense base over there, but for peaceful researches, any country can visit Antarctica. So in 81-82, our India has visited that region, and particularly it comes on the Norwegian region. You can see that on the east, it is Australian region, then it is the British, USA. So there are a claim of over 7-8 countries within Antarctica before 1959. So after that, International Geophysical Year, there was a treaty, and within this treaty, it includes all 14 articles that we will go for peaceful researches, no defense base over there. So Antarctic researches is started with different other countries too. So now we have more than 70 countries which are member of this Antarctica Treaty, and for that membership you have to work scientifically over there, establish some centers, then only you can get this membership. So what happened after the downfall of Antarctica of Russia, the Russians, the Germans which are situated near one, I will tell you something how this looks up. In the year 83, wintering this Dakshin Gangotri is there. So 1983, then status of India. We got only within a year the status of our membership because we have carried out a large number of meteorological studies over there, and a large number of international magnetic data we have to produce, India has to produce, because the German station which was based on Russians, it was closed because of the downfall of Russia. So most of the meteorological data, most of the geomagnetic data which are used internationally, they are produced by Indian station. So we got that membership quite early. Then we have got a large number of these membership of different communities. Then in the year 1988-89, second permanent research in Maitri was built, because by fault the first Dakshin Gangotri station was built at the near the sea, where there are frequent snow blizzards, and it covered that station completely. So it gone 40 feet down, because in Antarctica the snow blizzards are frequent. Sometimes after it continues for 10 days. So it covers whole the building. Whatever comes in front of that snow, it covers on one side. And when this Dakshin Gangotri station was defunct, before that they have established this Maitri station in 60 kilometers away from that area in a rock outcrop. So you can see that within this Antarctica, our position is this one, where we situated. This particular map is called, in Antarctica, this is called Schirmacher Oasis. Schirmacher, there was a German who first established the laboratory, visited this area. So after his name, they have given this area Schirmacher Oasis. It is 20 kilometers it covers about 20 kilometers, Schirmacher Oasis, and on the glacier, near the glacier. On the downslide, you can see our research station at Maitri. It has 26 rooms. We have all four facilities over here. We have one doctor every time it visited during this expedition. Then we have greenhouses over there. Then we have good kitchens and all facilities are there within this structure. So as I earlier I told you that two type of participants are there. Sometimes some are summer participants like me who visited that area, who visited Antarctica only for 6 months. But there are winter participants who study there for whole year and they remain there for 16 months. They are called winter participants. So this particular place is erected or established for those winter participants to perform different experiments and to stay there. After Maitri, we have third research station. It is Lajarman Hills and it is towards the Australian claim, and we have established this third one in the year 2000, around 2000, they have established this research station. Now it is again 2000 kilometers away from Maitri. It is too distant from there. So any scientist who has some proposal can visit Antarctica, and before that means when we are selected, it is National Center for Antarctic and Ocean Research situated at Goa. They are responsible for carrying out this expedition every year. Selection of the team member, it means you should have a good project proposal. It will go for screening and presentation of proposal. If you are selected, then you will go for medical examination at AIMS, training at ITBP Oli. Means it is the place where you can learn about rock climbing and in Uttaranchal. Then lectures you have means you have to attend certain lectures which is regarded the survival of Antarctica and firefighting trainings and all. So in Antarctica, the relative humidity is very low. It's less than 40%, and plenty of oxygen is there. So what happens? The fire, these accidents are very common in that area. So this training is to know how to get that firefighting training in Antarctica as well. Then physical training, as I was talking about rock climbing and all, this is given by ITBP, Oli. So this is the route earlier. Earlier, we have to move from Goa to Antarctica, which took about a month to cover 11,000 meters. But nowadays, there is a direct flight from Mumbai to Cape Town, and they stay there, and from there, by air, it will take only 6 hours to reach over there. This is the common site. A special type of ships, they have a raised bow because it has to break the snow, and it is a common site that you can see glaciers, or these glaciers floating. You can see these penguins and these seals. So this is when I have visited. This is the big ship. This ship is MV Thuleland from Sweden. It was hired. Then a large number of these army personals, Navy particularly, they use these MiG MI helicopters, choppers, which we use from ship to transfer to the Maitri. So we studied there. These penguins, they frequently visit. Antarctica is called pulsating continent. Why? Because during winter, it covers by more than 1,000 kilometers by permanent snow. Means you can see that snow melts during summer, and during winter you can see that it accumulated all around Antarctica. So it's contraction and expansion. That's why it's called pulsating continent. From an aerial view, you can see that our research station, Maitri, you can easily see that there is a glacier on the back side. It is used for as an airstrip for choppers to come and land on, and even they go for bigger, smaller airplanes which are hired from Russia, from Cape Town to this place. You can see that this is water supply. There is a lake, Priyadarshini Lake, nearby. Jub Lake we called it earlier, but it's named after Indira Gandhi's Priyadarshini, and this is the water supply. It's a special one because it is snowed here. So first there is a heater inside that lake which melts the water. Then a warm pipe along with the water moves up to 1 kilometer to the station. Yes, again it's a aerial view, and you can see that for summer participants they provide these huts where we have to fix our experiments, we put our microscopes, and we stay on these huts too. Again, as I told you that Antarctica is an international park. Nobody can contaminate it. So whatever the human waste, we have to burn it. Even the human shit, we burn it. There we collect the ashes, and then only we throw it 60 degree south latitude, after 60 degree south latitude. So synonym of Antarctica is a continent of extreme, icy continent, frozen continent, land of penguins and midnight sun, residence of lizards, seventh continent or white continent. We all know about how origin of Antarctica is there. It is the continental drifting. Means if a biological science student can easily understand, when there is cell division, there is a phase, we call it metaphase state. Or after that comes the telophase. Means it is similarly the chromosomes, they start moving towards the pole. Similarly, all over these continents, the northern ones, they moves upward, the southern ones moves downwards. So it took crores of years, and this Antarctica is separated. Then, why day and night for 6 months? Means you can see the position of Earth. It is based on the base of that one, and only for 6 months the sun is visible. So for 6 months there is no night over there. Continuous day is there. So we visited that one during the day period in Antarctica. Now one question, why Antarctica is considered coldest? Means the coldest temperature over there is minus 89.6 degree at Vostok. There is a place, Vostok, in the center of Antarctica, and there the temperature is minus 86.9 degrees centigrade, because it is situated on the base, and when the earth rotates, inclined sun rays, which has a small amount of shortwave radiation. This is one radiation is absorbed by atmosphere before it reaches the continents. Some are reflected by clouds. Most of the radiation is reflected back into the space by snow, because it's white and it is reflected back. Why Antarctica is windiest? There is no landmass. If you see that there is no landmass between South Africa and Antarctica, that peninsula, the neck. So no landmass is there. Less obstacles on the continent to block the wind currents. No plants, no hills. Why Antarctica is dry? No humidity. As I told you, it's less than 40% of this relative humidity is there. Very less amount of water in liquid, and direct transfer of water molecules. Why people go to Antarctica? Yes, they can go. Nowadays, they can go for adventure, commercial activities, and research. Third, whaling and sailing. Earlier, the discovery of Antarctica was based on whaling. And when I visited Antarctica, that was Operation Krill. There is a small squid-like animal is there, and it is interesting because this krill goes Antarctica to breed. It produces eggs there, while it lives in the coastal regions or in the center or warm regions of sea. It moves to Antarctica, lay its eggs. They go to the bottom, and along with cold currents, they move towards this warm areas of sea. So particularly one scientist from National Institute of Oceanography studied the movement of these krills or the life cycle of these krills in that particular expedition. That's why our expedition was Operation Grace. A large number of minerals and other natural wealth are there: gold, graphite, silver, coal, iron. But due to this Antarctic Treaty, exploration or mining of these minerals is prohibited. Now, research scope at Antarctica is a heaven for scientists because it's a full of problems and challenges are there. Continent of unexplored and unrevealed mysteries. Continent of unique phenomena, and it is declared as World Park. Nature's open laboratory is there. Means the climatic conditions and it's a cold, but it's a free from bacteria and all. Particularly sometimes the bread or sometimes food articles which are carried in 10 or 20 years before, they are lying there without damage. But some color is gone. For example, the haldi color or yellow color, it vanishes, but the chips and or other chapatis or other food items, they are remaining same without any damage or disposal or damage from this bacteria or some pathogens. So what are the different areas of research fields? Meteorology, yes, the IMD Department of Institute of Meteorological Department, Delhi, they are working on. Geomagnetic Institute of Geomagnetism, Colaba, Bombay, they are working on this. Glaciology, Geology Department, they are working. Upper atmospheric science, it is how this ozone depletion is there, and what about the environmental science, even pollutants are there, or what are the different metals are there, geophysics, biology, and paleontology. So these are the different areas where you can pursue researches in future in Antarctica. So I will tell you something about ozone. Ozone, we all know that it is particularly the higher concentration in the troposphere in human respiratory, and it causes chest pain. We all know about that. Ozone layer is stratosphere filters UV. What happens if the industry, the electronic industry, earlier these CFCs and all these chemicals, they were used, aerosols or whatever, and they were discovered in the year 1928. The British Antarctic expedition scientists, they observed in 1980s that a area about a football ground is depleting with ozone. So what happened? Whichever we have produced 100 years before, it reaches Antarctica in about 100 years. How it moves? We all know that whatever we produce, these CFCs or these gases which are responsible for depletion of ozone, they accumulated at the equator. Then there is a polar vortex, we call it. This polar vortex, it stretches the chemicals which present in center to 25 kilometers above in the equator. It moves towards the pole. At pole, what it does, it reacts with. We all know that the formation of ozone in nature is there, but the speed of destruction is higher than the product formation. So this oxygen, in presence of sun, it gets into oxygen O and O. Again, this oxygen again comes in close with this O and it forms ozone formation. This is the particular which is going on nature. This is the reaction which is going on nature. But this X is greenhouse gases, which we produced 100 years before. What happens when it reacts with ozone? It breaks the ozone into oxygen and O molecule. It's with. Then again, further it joins with this O and molecule and releases oxygen in free state. So a single molecule of greenhouse gases is responsible for addition of about 1,000 of ozone molecules. So this is going on in Antarctica. Auroras, they are common. We all know that Earth has a ionized, Earth is surrounded by ocean of ionized gases and charged particles called plasma. Sun has its own plasma, when certain gases, when they react with this plasma, sun also has plasma, interaction of solar winds with Earth's magnetic field causes emit to protons. And the gases which present in that atmosphere, they produce different colors of these clouds, and we call them auroras. Depending on the color, it has aurora australis, aurora new zealandis, and is a common feature in Antarctica. If you see the biodiversity, there are 7 species of penguins. We all know that. Throughout the world, there are 17 species of penguins, of which Antarctica has 7 species, 6 species of seals, 9 species of whales. And if you see, only 2 flowering plants are known from Antarctica. One is Colobanthus quietensis and other is Jessentia antarctica. They are the recent inhibitors. Long back, in earlier expeditions, there is a lot of these animals they were carried there in Antarctica, and along with those animals, some grasses for as their food or as or for their warmth, it was carried, and these grasses are recently colonized over in some areas, and only in Antarctica you will find only 2 of these angiosperms. Yes, we have large number of algae there, 700 aquatic and terrestrial, 700 seaweeds, fungi again hundreds, bryophytes 130, and about 400 species of lichens. As is this most common penguin there is emperor, which is 4 feet big, and Adley, which is only 2 feet of height. Then we have 6 species of seals, and they are named based on their some structure, mouth structure. Elephant seal, they have a long trunk-like elephant. Then we have some leopard seal, which has leopard-like mouth, and Weddell sea or raw sea, they are named after the seas on which they occur. Then 10 species of whales. We all know about this blue whale, fin, southern right, hempwhale, oreca, southern bottlenose, sperm, and south foot, 4th foot. 1 species of dolphin. Biodiversity of Antarctica, 35 species of birds are there. Albatross, the birds having the largest span of their wings, it is there. 120 species of fish. Then krill, I was talking about, and 66 species of insect. And you can see the view of different birds. It's this penguin, this emperor, LD, king, all penguins, 7 are there. Then it is common this, we all know about the scalps. Scalps are large source of iodine, and we will find plenty of this scalp in Antarctica. Then bryophytes, mosses, as I told you, only 20-30 liverworts are there. Then Arctic fungi, 30 species of macrofungi, 70 species of microfungi, and how they adapt, they increase intercellular trioles and polyols, and yes, 450 species of lichens, a total of 80% of lichen species endemic to Antarctica. We do not find them here in India, but still we have studied that how this, because earlier Antarctic part or this part, south part was associated with this, our Indian part. So we have studied the association of lichens, Himalayan lichens and Antarctic lichens, and we have recorded that a good number of lichen species, particularly one genus, yellow-green genus, yellow-red genus of lichens, Stylostasias lichens, they are common in Antarctica and our Himalayas also. You can see that this one, this orange-red color of lichens, they grow in Antarctica as well as in Himalaya. Most of these species are there. This is a common and mosaic of lichens on floor. Even you can see hanging them. Even you can see that they have to develop one cushion, and this is particularly which we use for lichenometry, and these are common Antarctic lichens. Thank you. Hello?
Hello?
Yeah.
Thank you very much, Dr. Opreti, for your wonderful talk and taking us to the journey of fascinating life of lichens. I will now request Dr. Sarvatra to take over and give the vote of thanks. Thank you so much. Over to you, sir.
Thank you, Dr. Aditi. Thank you so much, team VSP NDRI. Indeed, it's a great pleasure for all of us to listen our both the dignitaries, Honorable Speaker Professor Padam Shri Professor Pramod Tandon sir, Dr. D.K. Preti sir, and all the viewers and listeners, those who are participating in this virtual mode today's event, for get benefited by the economy and protection of the bioresources of our country. It is really a great pleasure and privilege to all of us to listen both of you, sir, and not only CSR NBRI on behalf of Office of the Principal Scientific Advisor, Ministry of Culture, Vigyan Prasar, CSIR, and particularly CSR NBRI. We are grateful to both the speakers. We are conveying sincere thanks on behalf of all the organizers to both of you, sir. And I hope all the listeners got benefited by both the lectures, bioeconomy part of our bioresources as well as the bioprospection of the beautiful world of lichens. So thank you so much, Professor Tandon sir and Dr. D.K. Preti sir. Over to you, Aditi.
Thank you.
So thank you, sir. With this, we came to the end of today's talk session. We'll be taking a short break and we'll assemble afterwards for the screening of science-related movie. Thank you all, and hoping to see you again soon in the concurrent session. Thank you again. Thank you so much.
Thank you.
In this garden soil. Soil is a magician. If one wants flowers, it gives of so many kinds. beans and corns, how wonderfully all are formed. Some bless us with aroma oil. There must be some magic in this garden soil. It shows big tricks, raises tall trees on its palm, with their roots rigidly fixed. Winds try to topple them hard, but they're not spoiled. There must be some magic in this garden soil. When we walk with nature, we receive far more than we seek. Let us take a deep breath and get ready to step into the homes of amazing plants. Let us walk with a sense of wonder, which sparks the journey of science. This garden is a special one. We are in a world-renowned botanical garden of Council of Scientific and Industrial Research, National Botanical Research Institute. Botany is a field of science where the magical world of plants is demystified. Spread over an area of 25 hectares in the heart of Lucknow, the capital city of Uttar Pradesh. It is one of the oldest and the third largest botanic gardens of India. CSIR's NBRI is home of scientists, technologists and gardeners passionately committed to the cause of botany.
The Botanic Garden of CSIR NBRI is unique in at least three aspects. First is the conservation status. The Botanic Garden of CSIR NBRI conserves about 6,000 different species belonging to different taxa and families. And the most exciting feature is the conservation in houses. Besides the conservation of plant varieties and species in the field gardens, we have specially designed plant houses in which we maintain specific temperature and humidity conditions for a particular group of plants. So this unique feature is not seen in all other gardens. So the conservation in houses is special. We have moss house, we have fern house, we have house for cactus succulents, palms, orchids, bonsai. We have a Jurassic gallery which is very interesting for these young students who can see the plants belonging to the Jurassic period. The conservation in houses is unique, and then also we have a research component linked with the conservation. We work on propagation of plants, we study the reproductive behavior of the plants which are difficult to propagate, we try their tissue culture, and simultaneously we work on the genetic improvement of ornamental plants. We try to introduce the wild ornamentals into domestication. So this particular is the second unique feature of our botanic garden. Then third is our connect to the society, that is known as the extension, outreach, education activities. We have some interesting thing for all the age group of our country. For example, children basically up to 8th class standard, they do not have idea about the plants which they use in their everyday life, what they eat, the fruits, the vegetables, the cotton plant, the sugar cane, from where we get the sugar. So all these plants we have introduced in section known as Know Your Plants. So this is basically introducing the importance of plants to the students, which are the plants which are used in everyday life. We also organize number of training programs to promote the interest and activity of gardening and related things, like people are interested in home gardening, in bonsai making. We have training programs for almost all the segments of the society, for young students, for housewives, elderly people, senior citizens, and also for the researchers. So these programs basically aim at various gardening related techniques which not only promote healthy living but also provide opportunities for income generation. So these programs are very popular. We have offline and online modes. Besides this, a lot of students of all categories, of all colleges, they visit our garden and enjoy the diversity that we conserve in this botanic garden.
Succulents House has a very rare gymnosperm member, tree tumbo, from the coastlines of South African Namib deserts. Touch and Smell Garden is first of its kind in the Indian subcontinent and 7th in the world. It is meant for enabling visually challenged Vigyang persons to learn about the diversity of plants, as they have a better sense of touch, smell and hearing ability. Ficus House and Navgreh Vatika are the latest additions, and Moss House holds a special place. Let us know our plants and be friends with them. Let us see them grow. Let us play with them. Now we are ready to see the beauty of elegant flowers in their heavenly habitat. Botanic Garden is rich with the presence of extensive varieties of bougainvillea, canna, chrysanthemum, gerbera, gladiolus, hippostrum, Nilambo lotus, rose and tuberose. Lake and lotus pond are bestowed with such a great charm that regular visitors feel enchanted near them every single morning. Rose and gladiolus show in January and chrysanthemum and coleus show in December. Two festivals are celebrated annually to spread aesthetic sense and awareness among the countrymen. Is the result when the divinity of evolution melts with the elegance of nature to amalgamate into beautiful feature. Let us witness the grandeur of some giant trees which appear inviting every guest to meditate in their hospitality. There are 300 kinds of trees in the Arboreta. Parijat tree and banyan point are the abodes of serenity in this botanic garden. If you aspire to follow the path led by eminent scientists, or walk on the tracks of NBRI Botanic Garden named after them may ignite your passion. Pilgrimage, one has to tread on the uncharted path with the quest to know till the pinnacle is reached. NBRI Botanical Garden truly mirrors the spirit of Indian people and culture, where diversity coexists without conflict and spreads the message of aesthetics, harmony and peace with a gaze towards a sustainable future. For there is peace on earth, peace in plants, peace in trees. Let us pray for the whole existence is peaceful.
Yes.